Hello, and welcome to today's webcast, the Biosimilars Update. My name is Bev Bromfield, and I'll be your moderator for today's presentation. We've taken a screenshot of the attendee interface. You should see something that looks like this on your computer desktop in the upper right corner. You've joined this presentation using your computer speaker system by default. This means if you hear music through your computer, you should be able to hear the presentation. If you prefer to join over the telephone, select Use Telephone in the audio pane, and the dial-in information will be displayed. You'll have an opportunity to submit questions to today's presenter by typing into the question pane on the control panel. We'll collect these and address as many as possible during Dr. Yamauchi's Q&A session today. In July, the National Psoriasis Foundation launched a new strategic plan which guides everything we do as an organization. To achieve our mission, the following goals were identified: lead collaborative and transformational research in psoriatic disease, improve health outcomes by increasing the lifespan and health of individuals living with psoriatic disease, secure resources necessary to achieve our mission-related goals. By attending today's program, you've already taken a step towards expanding your knowledge about psoriasis and psoriatic arthritis, moving towards a better understanding of what it means to live with psoriatic disease. If you have psoriatic disease, you can help accelerate our efforts towards collaborative and transformational research that leads to a greater understanding of the disease and a cure by participating in the NPF Corona National Psoriasis Patient Registry, which is the largest independent observational registry of psoriasis patients in the U.S. This registry represents a network of dermatology clinics that collect and study patient health information, allowing researchers to compare the safety and efficacy of psoriasis treatments, better understand conditions that are related to psoriasis, and explore the history of the disease. You can share your experience with researchers about what it means to live with psoriasis and psoriatic arthritis by participating in Citizen Scientist. Answer questions and view your data, along with over 4,000 participants. Data is used to help researchers gain a better understanding of the disease. To learn more and participate, visit the websites listed on the screen. We continue to improve the health of individuals living with psoriatic disease through our Patient Navigation Center, the world's first personalized support center for people impacted by psoriatic disease. The Foundation's Patient Navigation Center is available to help you find physicians, treatment information, navigate insurance issues, access support through our one-to-one program, and more. Contact us at 1-800-723-9166, option 1, or visit psoriasis.org/navigation center. To help the Foundation achieve its goals as mentioned, please support our mission through donations or by participating in local Team NPF events near you. You can learn more at psoriasis.org/donate or teamnpf.org. Now I'd like to introduce you to our speaker, Dr. Paul Yamauchi. Dr. Yamauchi is a dermatologist in practice with the Dermatology Institute and Skincare Center and Clinical Science Institute in Santa Monica, California, where he specializes in general dermatology and hard-to-treat issues such as severe psoriasis, offering the latest advancements and comprehensive care. Dr. Yamauchi is also a clinical assistant professor of dermatology in the Division of Dermatology with David Geffen School at UCLA Harbor Medical Center. Dr. Yamauchi is a principal investigator in several clinical trials, including psoriasis, skin cancer, eczema, and acne. He is widely regarded as an outstanding clinical research investigator and has served on advisory panels designing protocols that led to FDA approval of various medications currently not available on the market. The Clinical Science Institute has been conducting clinical trials for over 20 years. Dr. Yamauchi is a past member of the National Psoriasis Foundation's Medical Board. He also serves as the President's Council member, which is an elite group of healthcare providers committed to the mission of the Foundation and finding a cure for psoriasis. Dr. Yamauchi is widely regarded as a key expert leader in the field of psoriasis and has extensive experience with use of the latest biologics, oral drugs, and topical treatments. It is my honor to welcome Dr. Yamauchi as today's webinar speaker. Please welcome Dr. Yamauchi.
Great. Thank you so much, Bev, for that wonderful introduction. And hello, everyone. And it's a privilege and an honor for me to do this presentation on biosimilars. So what are biosimilars? That'll be the focus of today's discussion. And for those of you who are listening, you may have moderate to severe psoriasis and you may be on a biologic agent already to treat your psoriasis. I have to say, for the past 15 years, biologic agents have been a game changer for many people with moderate to severe psoriasis. And my goal to treat psoriasis is basically for you to forget that you have psoriasis. You're not itching, the plaques are not visible, and I want you to get to as clear as possible so that you can do the things I take for granted, like going out to the gym in shorts or going out to the beach and not having to worry about having psoriasis. So what we want to discuss today is the difference between a biologic agent that's brand name versus a biosimilar, and then we'll go over the pathway in terms of how the FDA approved the initial biosimilars in the United States. And we'll talk about the safety and efficacy of biosimilars as well as implications of using this to treat psoriasis and other inflammatory conditions. So biosimilars are new. We have no experience with biosimilars in the past, but there are some concerns for the dermatologist in terms of biosimilars. Number one, would they be effective as the original biologic agent? Will be as safe as the original agent? And what about substitution? Let's say that you're on a drug already for your psoriasis. Let's say Humira, for example, and it's under good control. Would a pharmacist be able to substitute Humira for a biosimilar in the future? And if so, will that affect you in a negative way? But the main goal of biosimilars is to reduce the cost of biologic agents so that these kinds of agents can have better access to our patients. So that's the main purpose, is to reduce the high cost of biologic agents. Between 2012 and 2018, in that 7 out of the 10 prescriptions worldwide are biologic agents, with Humira as the number one drug prescribed in many conditions such as psoriasis, rheumatoid arthritis, Crohn's disease, etc. So it is the most widely prescribed medication. And you can see other drugs that you might be familiar with, Enbrel or Remicade, which are also approved for psoriasis. We have several biologic agents out there for psoriasis, 11 actually, so it's a very crowded market. And I just listed a few of them. You probably will recognize these names: Humira, Remicade, Stelara, Cimzia, Enbrel, Cosentyx, and Taltz. And what I've listed is the expiration date in the U.S. for these different biologic agents. So Humira already, their patent has expired 2016, but the first biosimilar for Humira won't be available until 2023. Remicade already has a biosimilar. Generic name is called Inflectra, and to date, there are 3 biosimilars to Remicade that I know of. In dermatology, actually, we don't use a whole lot of Inflectra or Remicade to treat psoriasis because it is an IV infusion. But rheumatologists as well as gastroenterologists use a lot of Remicade to treat rheumatoid arthritis and Crohn's disease. And you can see that the other biologic agents, the expiration date for the patent won't be until the 2020s or even in the 2030s. So the first biosimilars already available are Humira, Remicade, and perhaps Enbrel. As you know, biologic agents are very, very expensive. It can range from like $50,000 a year to maybe $100,000 a year. The reason why they're expensive is because of the high manufacturing costs associated with biologic agents. And they're more expensive than oral pills. However, even some of the oral pills can be quite expensive, and the price can be $30,000 or more for some of these oral molecules. But nonetheless, we know that these drugs are expensive. The most expensive drug right now is one that's $1 million for a very, very rare condition in infants. So the goal, as I said before, of biosimilars is to reduce the cost of biologic agents and for better access to more people. So the BPCA Act, in March 2010, President Obama signed the Biologic Price Competition and Innovation Act. And what it is an abbreviated licensure pathway to fast-track the approval of biosimilars or make them interchangeable relative to biologic agents. I'll explain that more in detail later in this talk. This act was similar to the 1984 Hatch-Waxman Amendment, creating the generic drug program for small molecules. So what are biosimilars? So number one, what is a biologic agent? A biologic agent like Humira or Cosentyx or Taltz is a protein. Usually they're in the form of a monoclonal antibody that targets the immune system. And that's why it's a shot, not a pill, because it's a protein. If you were to consume it, it would be digested in your stomach acid. So that's why it's given as a shot. So a biosimilar is a molecule that's very similar to the brand name biologic agent, but it's not identical. There's some differences between a biosimilar and the reference biologic agent in terms of the molecular structure. They're very minor differences, but they're not exact copies. So number one, it has to be similar to the reference biologic agent, and also no clinical meaningful differences between the reference biologic agent and the biosimilar in terms of safety, purity, and efficacy of the product. Biosimilars and biologic agents are made by the same process. So what happens is that through DNA technology, you take the DNA of that molecule that you want to replicate and you insert it into a cell. And the cell in turn multiplies and replicates and it forms from the DNA the biologic agent. And these cells are grown in big cell culture, almost like a beer factory in this huge vat where the cells are maintained and grown. And then they produce the biologic agent or biosimilar, and it's finally isolated and then characterized and then finally made into the product where it's refrigerated and then it's given to the patient. So anytime there's this process that if one of the vats gets contaminated, then you got to throw the whole thing away. So the quality control of manufacturing biologic agents is very, very stringent and very strict and also a very expensive process. So biologic agents are larger and structurally more complicated than synthesized drugs. So if you see in this graph, aspirin on the far left corner, it's a tiny molecule. Insulin for diabetes is larger. It's about maybe 500 times larger than aspirin. But then you go down to, on the far right, a monoclonal antibody like Humira or Cosentyx or Taltz or Tremfya. They're huge molecules because they're antibodies. So relative to insulin, a monoclonal antibody is about 30 times bigger than insulin. So they're really quite big. So what's the difference between a biosimilar and a generic? They're not synonymous with each other, biosimilars and generics, although easy way for me to describe a biosimilar is simply put like a generic biologic agent, but that's not exactly 100% true. So the differences between a generic, like Lipitor, for example, or cholesterol, in that they're small relative to biologics and biosimilars, which are huge. So that's one difference. Number two, the structure. The structure of a small molecule aspirin is simple and well-defined, whereas with biologics and biosimilars, they're very complicated. They're composed of something called amino acids in a long chain, and they're also covered with sugar molecules and things like that. So they are very, very complicated molecules. To manufacture small molecules, it's easy. It's like organic chemistry. It's predictable, and exact copies can be made. But with biologics and biosimilars, since they're made by a cell line, there can be some changes to the structure of biosimilars and biologic agents. So that's why, while very similar, they don't have the same exact chemical structure. Small molecules are easy to fully characterize, but biologics and biosimilars, because of their huge nature, a little bit more difficult to characterize. Small molecules can be stored at room temperature, whereas biologics and biosimilars need to be kept refrigerated so that they don't destabilize. And then also something called immunogenicity, where you form antibodies against the structure. So with small molecules, you really don't have that problem of immunogenicity. But with biologic agents, you can form antibodies, your own antibodies against the biologic agent. If that happens, then the drug might stop working because basically you have immunized yourself against biologic agent, and then you might have to switch to something else because it can stop working. So I'm going to talk about how the FDA approves a biosimilar relative to a biologic agent. The pyramid on the left is for the reference product, like Stelara or Skyrizi or Tremfya, whereas the pyramid on the right is for a biosimilar development. You can see that there are four categories, same categories: analytical characterization, non-clinical, clinical pharmacology, and safety and efficacy. So we'll go over this individually. But the main difference is that the FDA cares more about the analytical characterization, the structure and function of the molecule relative to clinical investigation studies. So here, analytical characterization. So what you want to do is to identify the structure of the molecule, make sure that it's relevant in terms of the relative to the reference product in terms of analytical methodology, the structure, the function, etc. So FDA places more emphasis on this with the biosimilar relative to a biologic agent. Non-clinical studies include animal studies to make sure that there's no toxicity, and that has to be done for reference biologic agents. But if the biosimilar has been designated to be very similar to analytical characterization, then non-clinical studies like animal toxicity data are not really necessary. Clinical pharmacology is something that has a lot of emphasis both on the reference biologic agent and the biosimilar. What this means is when you inject the medication into your body, how does your body metabolize it? What are the levels of serum concentration of the product? So here, the FDA requires that the amount of medicine in your body, called pharmacokinetics and pharmacodynamics, is similar, has to be similar between the biosimilar and the reference product. And finally, we have safety and efficacy, the clinical trials, the phase 2, the phase 3 trials. So for the reference product, like Humira, which has 10 different indications, the company has to do 10 different trials for safety and efficacy for all the different indications, whether it be for psoriasis, rheumatoid arthritis, Crohn's disease. But for a biosimilar, all they have to do for FDA requirements is just do 1 or 2 trials in any of the diseases. For example, if you do a trial on a biosimilar for psoriasis and the efficacy and the safety has been demonstrated to be similar to the reference product, as well as the analytical characterization and the pharmacodynamics of pharmacokinetics, then the company that makes a biosimilar is not required to do trials for other indications. So if a product met similarity to psoriasis, then that biosimilar can be approved for Crohn's disease or rheumatoid arthritis, even though it was not studied in a clinical trial. So that's the main difference, is that for the reference product, they have to do trials for all of them, but for biosimilar, they only have to do like 1 or 2 trials to demonstrate similarity to the biosimilar. So extrapolation, so I just sort of described that right now. So extrapolation is where you do a trial, let's say in psoriasis for that biologic agent. If it succeeds, then that data in terms of safety, efficacy, analytical characterization is extrapolated to other conditions. For example, with Humira, let's say that the biosimilar was tested for, let's say, psoriasis and rheumatoid arthritis, then that biosimilar can get approval for other conditions like Crohn's disease or maybe in kids with Crohn's disease and other conditions. So that's the main difference. And the reason why extrapolation is done is to save cost, because it's very expensive to conduct clinical trials. So for biosimilar to reduce cost, they can only have to do 1 or 2 trials, and the data and the indication is extrapolated to other indications. Interchangeability. So that's the issue that we're sort of concerned about, is that substitution. Can one reference product like Humira be substituted for a biosimilar? Currently, none of the biosimilars are interchangeable yet. That could certainly change in the future. So at the moment, if a person is on a brand name biologic agent, the pharmacist cannot automatically substitute it for a cheaper biosimilar without the prescribers and the patient's consent. Right now, none of them are interchangeable. To prove interchangeability, what the company has to do that produces the biosimilar, they have to do switch studies. So they have to go from a reference product to a biosimilar and do that back and forth, switch between the biosimilar and the reference product back and forth to make sure that the efficacy is the same and also it's safe to interchange between the reference product and the biosimilar. So the FDA give the designation that the biosimilars can be substituted or interchangeable, but it's going to be the state laws that determine whether substitution will be automatic or not. So even though the federal government will approve interchangeability, it's up to the state laws to determine how interchangeable they can be, whether automatic substitution is allowed or not. So what are some of the guidelines for state laws regarding biosimilar substitution? So number one, any biosimilar under consideration for substitution must first be designated as interchangeable by the FDA. None of them are interchangeable. The prescribing healthcare provider can blunt or prevent substitution by writing dispense as written or brand medically necessary. Prescriber must be notified of the substitution by the pharmacist as well as the patient. And the pharmacist and the physician must retain records of the substituted biosimilars. And also, maybe some state legislation require that the pharmacist explain the price difference between the reference product and the biosimilar, and that the substitute product, the biosimilar, must have a lower cost. So this is the map of the United States showing the legislation on a state-by-state basis on biologic and biosimilar substitution. So this is the most updated map that I found back in 2018. So the green states have laws already enacted, and then the red ones are filed bills. So you can see that the majority of states in the U.S. have already enacted some legislation on biologics and biosimilar substitution. How do you name these things? So there's FDA guidance for non-proprietary naming of biosimilars. So there's two ways to name a biosimilar. Number one, you have to give it a brand name. So one of the biosimilars to Humira is called Amjevita, which is made by Amgen. Okay, the scientific name of Humira is called adalimumab, but to prevent confusion between the scientific names between the brand name and the biosimilar, the FDA required that you add a suffix to the scientific name. So the scientific name for Amjevita is called adalimumab ATTO, and that suffix will be different for another biosimilar that's going to be similar to Humira. There's also etanercept or Enbrel biosimilar called Erelzi, and the suffix for etanercept is SZZS. So the European Union have been way ahead of us in terms of approving biosimilars since 2006. So 2006, the first biosimilar was made called somatotropin, and then erythropoietin followed suit, and as well as other biologic agents for the past 13 years or so. And then for our relevance, Inflectra or Remicade was first approved in the EU in 2013. Also, the biosimilars for Humira are available in Europe, but not in the U.S. So it's gotten pretty good traction since the introduction. 2007, of course, it was low, but then over time, 2014 and beyond, that the uptake of biosimilars has been growing because of the reduced costs associated with them. So this is a list of currently approved biosimilars for psoriasis, and it may not be an all-inclusive list, but I think this is a pretty complete list. So you can see three categories: etanercept or Enbrel, adalimumab or Humira, and infliximab or Remicade. These are all TNF inhibitors, which you might have heard about. The ones that target other pathways like IL-17 or IL-23, since the patent expiration is still years or decades away, no biosimilar will be available for maybe 15, 20 years for the IL-17 or IL-23 inhibitors. You can see in the first column the brand name of these biosimilars, the generic or scientific name, that's with the suffix, and the dates that they were approved. So I'm going to give you an example of a biosimilar that demonstrated biosimilarity to Humira. So adalimumab ATTO Amjevita, it's also called ABP 501. So this is actually manufactured by Amgen in Thousand Oaks. They also make Humira. And so in July 14, 2016, the FDA Advisory Committee recommended approval of Amjevita. And what the basis of the approval was analytical characterization, clinical pharmacology, and 2 trials were conducted for Amgen-Beta, plaque psoriasis and rheumatoid arthritis, and I'll show you the data. But with these 2 trials succeeding, then Amgen-Beta indication for Amgen-Beta got extrapolated to other conditions, including psoriatic arthritis, ankylosing spondylitis, juvenile idiopathic arthritis, adult Crohn's disease, and adult ulcerative colitis. So that's what I meant by extrapolation. You just do a couple of clinical trials, and then you get indications for all these other conditions without having to do specific trials on these conditions. So this is an example of analytical characterization, the binding of Humira, both from the U.S. and from the European Union. That's what the FDA required, versus in the middle, Amgen-Beta. You can see that it binds to TNF very similarly. Okay, so that's analytical characterization. This is a slide looking at the improvement of the psoriasis called the PASI percent improvement. So PASI is a clinical tool that measures the severity of your psoriasis based on how red, how thick, how scaly the plaques are, and also what percentage of your body is covered with psoriasis. So you can see that there are 3 lines, Amgen-Beta in the circle, and Humira, both European and U.S. You can see that the lines pretty much overlap with each other. So up to week 16, the overall improvement of the psoriasis was pretty much identical between the reference product Humira and Amgen-Beta. At week 16, a switch was done from Humira over to Amgen-Beta, and the switch shows that the efficacy, the improvement of the psoriasis was maintained from Humira over to Amgen-Beta. And this is an example of demonstrating biosimilarity for psoriatic arthritis. So here we're measuring the improvement of arthritis 20%, 50%, or 7% between Amgen-Beta or ABP-501 and Humira. You can see that the lines have very similar efficacy, that they almost overlap with each other. So these studies for both psoriasis and arthritis demonstrated clinical efficacy that was similar. And also the safety was the same between the biosimilar and the reference product in terms of infections and things like that. The safety profile was pretty much the same. I should also mention that for the biosimilar, the package information will be the same as the reference product. So if you have a package insert for Amgen-Beta, it's going to be pretty much identical to the Humira package insert. So what are some of the issues with biosimilars for and against? So for, biosimilars are as similar to reference as reference is to reference, meaning that a biosimilar will be just as identical to the reference as 2 reference products. But then some people are arguing that they're not similar enough and variations can lead to changes in safety and efficacy. Studies being conducted in psoriasis will be adequate proof of biosimilarity. Some people may argue that you need to do studies in other conditions to establish biosimilarity, but to reduce costs, that's why the FDA just required only a couple of studies. Against argument is that switching may lead to immunogenicity. So if you switch from a reference product over to a biosimilar, could that induce antibody formation and would the drug stop working? And that is unlikely, but certainly a theoretical possibility. If widely adopted, biosimilars will increase access and decrease costs. And that was the main focus by Congress and President Obama to do this initiative. But then people are arguing it may not lead to cost reduction due to contracts with payers and the pharmaceutical company. 4, healthcare provider will have control over switching. But the against is that maybe not. Maybe healthcare providers may not have exact control, and it's a pharmacist that will have control on what the patient will be getting, whether it be the reference product or the biosimilar. Biosimilars will lead to significant cost savings. So a RAND study was done. If all patients were transitioned to biosimilars, spending biologic agents will fall between 10 to 50%. So cost savings of $44 billion over 10 years. Disagree? People might argue that, hey, that's because of the contractual relationship between managed care and the sponsors, that perhaps biosimilars may not be essential if the contracting continues and biosimilars are not part of the equation, so that only brand name products will be available. But again, that remains to be determined about the contractual relations between payers and the sponsors. So again, this is to summarize the important considerations while evaluating biosimilars, the clinical data. So I showed you the data for Amgen on psoriasis and arthritis. You saw that the efficacy was pretty much the same as the reference product, and also the safety was the same. Immunogenicity is still a concern, particularly from an FDA's point of view, that if you are going to substitute or switch, could that lead to antibody formation and decrease the efficacy of the drug? Product characterization is what the FDA really emphasizes on, the analytical characterization of these drugs, as well as product stability. The big question that remains to be answered is, at state level, will automatic substitution occur or not by the pharmacist or by the payers? And our healthcare providers, are they really going to be satisfied that doing a trial, the indication we extrapolated to other conditions? And finally, product naming. I've already mentioned that there'll be a brand name product for a biosimilar as well as a suffix for the scientific name. So that concludes my presentation, and I thank you for your attention. I think that hopefully in the next decade, that if the biosimilars are on board, that they'll be adopted. Again, the main goal is to reduce cost savings as well as better access to all our patients. So thank you very much for your attention.
Thank you, Dr. Yamauchi. That was a great presentation and very informative update about biosimilars. It's good to hear the latest information since they were first announced, at least in the U.S. in 2016 when the first approvals came. We'll now open this up for our Q&A session. If you have any additional questions based on what you've just heard, please submit them through the questions panel on your attendee interface. We'll attempt to answer as many questions as possible in the remaining time. First question, if I'm on a biologic and switch to a biosimilar but don't do as well, is there any reason why I can't go back to the biologic?
That's an interesting question in that if you are in a reference product and then you switch to a biosimilar, you don't do as well, can you go back on the original product? The chances of that happening are probably not high, that you should maintain the efficacy. But for some reason, if you don't experience the improvement, if you're not satisfied with the biosimilar, then yeah, you're more than welcome to have your doctor or healthcare provider switch you back to the original agent that you're on, and hopefully you'll be able to recapture that same degree of improvement. So certainly going back to the original one would be advisable if the patient is not satisfied with the biosimilar.
Another question, do the biosimilars require a TB test, such as the biologics? And if it comes out positive, what are your options?
That's a great question. So all biologic agents for psoriasis require or TB evaluation before you start it. And so that also will be applied to the biosimilars. So biosimilar to Humira or Remicade, you do have to have a tuberculosis test done at baseline. And I tend to check that on a yearly basis to make sure you have not been exposed to tuberculosis. In the event that your tuberculosis test comes back positive, then the next step that needs to be done is to obtain a chest x-ray. If the chest x-ray shows that you have active TB, change in your lungs, then you should not be on the biologic agent until that active TB is treated and in remission. But if the chest x-ray is negative, no signs of active TB, what that means is that with the positive test for tuberculosis, you have been exposed to it. Let's say maybe on a plane, someone was coughing next to you, and then you got the tuberculosis bacteria and you were exposed to it. And so in that regard, highly recommended that you take tuberculosis treatment with a pill for about 6 to 9 months, regardless if you're going to go on the biologic agent or not, because there's about a 10% chance if you have been exposed to tuberculosis that at some point you might develop active tuberculosis.
If you are coming off of the biosimilar or the biologics due to tuberculosis, what would your treatment options be?
So if you, let's say you were on a biologic agent or biosimilar, and all of a sudden you do the blood test and it came back as positive, then you do a chest x-ray. If that's negative, then first of all, you have to stop the biologic agent. And then if the chest x-ray is negative, you start taking these pills for tuberculosis for 6 to 9 months. But 1 month after you start taking the pills, then you can go back on your original biologic agent or biosimilar. So as long as you're taking the pills, if you've been exposed to tuberculosis, you can restart the body agent 1 month after starting the pills.
That's good to hear. I was afraid you'd be off of it completely.
Yeah, you don't have to be off it complete unless you have active tuberculosis, then don't be on it. But if you've been just exposed with no active TB, then you only have to stop it for a short period of time.
Another question, how do elderly people do on biosimilars? Are there any differences or cautions you need to watch for?
So in terms of biosimilars and biologic agents for elderly people, in my experience, and it's also been in the literature, that there's actually not a whole lot of difference in terms of safety between a older patient, say in their 70s, versus a younger one in their 20s and 30s. But if a older patient has a history of chronic infections, bad diabetes, history of several cancers, then there probably won't be good candidates for biologic agents. But in general, biologic agents are very safe, especially with the new ones that have come out in the past years. No cancer warnings, no serious infections, doesn't affect the liver, kidneys. For the most part, they're safe enough to use in the elderly population.
Another question has some concerns about safety. Is it safe to only do a trial on one or two diseases rather than all?
That's a good question in terms of safety, because the FDA put more emphasis on the analytical characterization of the molecule versus clinical trials. So far, in the trials that have been done for other biosimilars, that the safety was very similar to the reference product in those trials. So my opinion is that based on the fact that if the analytical characterization is the same, the pharmacodynamics are the same, and the clinical trials show that the safety and efficacy are same, just based on those one or two trials, then I feel comfortable that the safety can be extrapolated to the other indications.
And do the biosimilars have the same side effects as the biologics?
Yes, so the side effect profile for the biosimilar will be the same for the original biologic agent. So Humira has precautions about side effects, and those side effects and precautions will be the same for the biosimilar.
And how long can you take Cosentyx for and what?
So Cosentyx is approved for psoriasis, psoriatic arthritis, and ankylosing spondylitis, which is a back disorder. It's been on the market for a few years now. It's what's called an IL-17 inhibitor. It's the first IL-17 inhibitor to be approved for psoriasis. Since then, Taltz has also been approved, and also a drug called Siliq as well. And so all of them have very good efficacy. All of them, for the most part, have similar safety precautions. And psoriasis is a lifelong disease. It's chronic, and there's no cure for it. So with any biologic agent, including Cosentyx, you're on it indefinitely. So even if your skin is clear, you're doing well, still do your shots as direct because you don't want your psoriasis to come back.
And this question is coming from Canada, and they've been told that due to protocol, they have to go on medication that is some of the older treatments to treat cancer. So I'm assuming that might be methotrexate, rather than be given a biologic. They're assuming this could be due to the cost of the biologics. They've been forced to use topicals. So they're just wondering if a biosimilar could be a potential option to counter this issue.
Yes, that's my hope, is that because of the high cost of the biologics relative to methotrexate, which is cheaper, that hopefully you can bypass that step edit of having to go on methotrexate and instead go on a biosimilar, which hopefully will be a lot cheaper than the original product. And that's the main focus of biosimilars, reduce cost savings and greater access for patients so that you can go on something safer and more effective than methotrexate.
That's all the time we have for our questions. Thank you, Dr. Yamauchi, for such an amazing webinar and for taking time to respond to the questions we received, which were all pretty amazing. We certainly appreciate your time today and for sharing your expertise about the development and status of biosimilars. It was a great update.
All right. Thank you so much, Deb, and I really enjoyed doing this presentation. Have a good night, everyone.
You too.
Thank you.
Okay, so in addition to today's webinar, podcasts are also available through Apple Podcasts, Google Play, Spotify, Pandora, iHeartRadio, or a feed service of your choice. You can access podcasts at the website listed here. After hearing about biosimilars, you might try listening to the episode about biologics via the link provided on this slide, or hear Dr. Freed address the emotional impact of psoriasis with episode 38. Upcoming podcasts address relationships and nuances to being a care partner on November 19th, as well as fashion tips with psoriasis. Contact the Patient Navigation Center if you still have questions, would like additional information about treatment options, finding a physician, or having issues with accessing treatments. Contact our Patient Navigation Center by phone, email, or live chat. Once you leave today's webinar, you'll receive a survey to provide feedback about the presentation. Tell us what you think. We value and appreciate your comments. Finally, you can access our webcast archive at psoriasis.org/webcasts. This concludes our presentation today. Thank you for attending.